go ahead and get going. The eyes and vision. Okay, again, what's happening is the light impulses from the outside, the external world, is hitting a receptor for one type of receptor or another. It's being turned into nerve impulses. The type of receptor, the pattern, is interpreted into the brain as to what we see. Okay? Same thing with the eyes, just as it was in the ears. You know, the bottom line is what's going back is this, action potential, and the brain is the thing that interprets the whole thing. Anyway, the eyes. It transduces light energy into nerve impulses this time. We only see a very limited portion of the spectrum, 400 to 700 nanometers. That's just, other animals will see a higher spectrum. Actually, if you have the lens in your eye, filters a lot of it. So if you have a lens implant for cataracts, excuse me, yeah. This day starts at 4:30 in the morning. So if you have a lens implant for cataracts, they don't filter the same. You see more into the UV range because you don't have that filtering built into that artificial lens. Okay, structures of the eye. Fibrous tunic. This is all in your book. Sclera, the white part of the eye. Cornea. Connective tissue, transparent, convex. Vascular tunic. Middle layer of the eyeball. Highly pigmented, supplies blood, it's immunologically active, helps prevent infections in the eye. Middle layer, the posterior portion of the eyeball is the choroid. The ciliary body is muscles. They hold the lens in place. The iris is the anterior portion of the vascular tunic, and we'll have a diagram up here. Pigmented cells, that's where you get your eye color. Internal tunic is where you're part of the retina. The retina is part of the internal tunic. It's where your photoreceptors are, neurons, connective tissue. The retina is where your it's part of the internal tunic, where your photoreceptors are, and the lens is not part of any of it. It's between the posterior and the vitreous chambers. It's held there by ligaments, the ciliary body. Now what happens is, when we do the visual sensory lab, the ciliary muscles allow the lens to either flatten or curve. And the ability of that to happen is your ability to focus, and especially focus on things that are near. As you age, the ciliary muscles get weak, and you cannot focus. It's called a presbyopia. And as my ophthalmologist said, if you're over 45 and you're not wearing reading glasses, you're fooling yourself. And boy, when I hit 45, I'm wearing glasses today, but I'm wearing contacts. I hit 45. He was right. So what I do is have contacts that are a diopter too weak. I make myself nearsighted. Still enough to drive, but then I can read with them. And as long as that's working, you're not cutting on mine.
You don't want to do bifocals?
No, I don't. I just don't want to fool with them. I like the cheap contacts, and I just want to fool with them. So here's your structures of the eye. Optic nerve coming in, central vein, the fovea centralis. We'll talk about the retina. The retina is where all your cones and rods are, your photosensors. Choroid's sclera is the white of your eye. Choroid's supportive tissue. Your conjunctive is on the outside. Here's your ciliary bodies. Here. And your suspensory ligaments. That's what holds the lens, and that's what pulls the lens or allows the lens to relax or flatten out, and this allows you to focus on objects near and far, and mostly on objects near. Cornea, pupil, lens, iris. The pupils are what allows the light into the eye. Those are what, when you have between the sympathetic, when you have your fight or flight syndrome, the sympathetic nervous system opens those pupils way up. You get as much light as you can into the eye to see as well as you can. When the situations pass, the parasympathetic system kicks in. And this is the difference between having something that you would expect to be excitatory is actually inhibitory. Sympathetic is inhibitory, it allows them to open it. Parasympathetic is excitatory, and it closes them. Anyway, we will get into these. Light passes through the cornea into the anterior chamber of the eye. Passes, so there's your cornea. Cornea, here's your anterior chamber. Then it passes through the pupil. And that opens or closes to let more or less light in. Just like the lens of a camera, your f-stop on a camera. That's what your pupil does. Passes through the lens, which can change shape to focus the image. Nearsighted and long, farsighted. If you're nearsighted, your eyeball is too long. I haven't gone over this with you, have I? Okay.
Your eyeball's too long.
Too long. Let me show you. Or let me try to show you. If you're nearsighted, if there's your lens, your eyeball is too long. Light coming in from the lens is focused right there, not on the optic disc at the back of the eye. And so everything's blurred, because what happens is they're starting to spread out. You want this right on the optic disc. So you put a concave glasses in front of it, and by the amount of the refraction and that, you put the light right back on the optic disc. That's how glasses work. That's how contacts work. When they do LASIKs, what they do is they cut the eyeball, and essentially when it heals, it changes shape enough that it allows that to focus again back on the disc. If you're farsighted, hyperopic, as opposed to myopic. Myopic is nearsighted, hyperopic is farsighted. Your eyeball's too short. So instead of focusing on the optic disc, it's focusing back here someplace. So you have a convex lens that brings it back to focus on the fovea or the optic disc, the back of the eye. If you have astigmatism, astigmatism, what you have is, instead of your cornea being nice and round, it's misshapen. So you need a cylindrical lens to take this and correct the astigmatism, and again, get the rays back there. And we will test them. When we do the lab, we'll test for nearsighted, farsighted, and astigmatism. But that's a misshaped cornea, as opposed to having the eyeball too long or too short. Iris can increase or decrease the diameter of the pupil. You have the circular muscles via the parasympathetic stimulation. Constriction is parasympathetic again. Dilation is sympathetic. Fight or flight. So we're back to here. In dim light, your pupils dilate. Normal light, they're smaller. Bright light, smaller. Aqueous humor fills the anterior chamber. It's secreted by the ciliary bodies, the muscles, the ciliary bodies that are attached to the lens. Drains into the sclerovenous sinus. The aqueous humor also carries all the nutrients for that part of the eye. You can't have blood vessels running all through your lens and cornea in that, or you'll be looking at them. So everything, all the nutrients in that in there are by diffusion through the aqueous humor. Drains into the sclerovenous sinus. Inadequate drainage leads to glaucoma, which is a buildup of pressure in the eye. The canal of Schlemm gets blocked for one reason or another. Aqueous humor, you have your canal of Schlemm here, your ciliary body. This is what's excreting the aqueous humor. You have your anterior aqueous humor here in your anterior chamber, your iris, your canal of Schlemm, which drains it and reduces the pressure. I don't know where they get these names. Schlemm must be a physiologist someplace. Light refraction. We talked about light refraction when we looked at the microscope. As the light enters a different substance, it bends. It's called refraction, and it's measured on a refractive index. Air has a refractive index of 1.
What has the refractive index of?
Air has a refractive index of 1. Your cornea, your lens, they have refractive indexes of like 1.3 to 1.4.
Is that what they measured when your eyes tested?
No. That's part of the prescription, but what they're actually measuring is how far back they need to change the focus. And then they correct it with the refractive index of the lenses. Changing the curvature of the lens, let's see, light passes through one medium to another advance. Curvature at the point of refraction can also bend the light. So that's how lenses work. That's how the lens of the eye works. As if the lens flattens and as you're trying to see something further away, the curvature bends, the refractive index bends, you're able to focus. As if you need more curvature to see something close, and as long as your ciliary muscles can handle it, you can focus. When you can't, you get reading glasses. Because you just can't do it. Changing the curvature of the lens allows fine control of focus. Now, refractive index, because the light's bent, Just like in the microscope with the refractive index, the image is flipped over and backwards because of the bending of the light. So light refraction, it comes in, and I think I said I put it on the optic disc. You need to be on the fovea centralis, and we'll talk about that later. But the light's coming in, it crosses over because it's coming in from different sides. It's refracted and sent back. That's what flips your image. I use a large format camera for taking pictures. Put the hood over your head. Let's speak right. And you've got to learn because everything's backwards. Yeah, when you're looking at it, and I'll when we do the lab, I'll bring it in and let you look through it. But it's the same deal, and you have to learn to live with it. Visual fields. You have visual fields. Your eyes split into visual fields. Each eye has two visual fields. And it's projected onto the retina. The right side is projected onto the left side of the retina. The left side is projected onto the right side of the retina. You can see where they cross over. That happens with your visual fields. Right, left, left and right. This is your fields of vision. They get sent to the different sides. They get projected on different sides of the retinas. The lens, it's suspended. It's hanging there. It's got the suspensory ligaments. It's attached to muscles called ciliary bodies. As they relax or contract, it changes the shape of the lens. It allows you to focus. So here you have your zonular fibers, suspensory ligaments, your lens, your iris, your conjunctiva, your cornea, your sclera, the outside, your ciliary muscles. These are your ciliary muscles. They go around the eye. As they contract or relax and they lose the ability to contract, you cannot focus. You get what they call a presbyopia, the inability to focus on things near, hence the reading glasses. Accommodation is the ability of the lens to keep an object in focus on the retina as the distance between the eye and the object moves. What you're going to do in lab is you're going to start with a yardstick when you've got a needle, and you're going to bring it back until you can't focus anymore. Fortunately, if I don't learn, my eyes are so bad that I don't think yours have anything. Mine,
Pretty bright.
if I on glasses, my diopters are 12 or minus 12. So before they had high index plastic, my glasses would literally be 2/3 of an inch thick. They were Coke bottles for max. God, I love high index plastic. Again, high index plastic refers to the refractive index. The high index plastic is able to change the refractive index of the light with a thinner glass or thinner plastic. Anyway, accommodation is the ability of the lens to keep an object in focus. It's a contraction of the ciliary muscles. It's good for close vision. If it works while you're young, enjoy it. Relaxation of the ciliary muscles pulls on the suspensory ligaments, causing the lens to thin. That's the distance vision. If they are too weak to contract, then you don't have the near vision. This is accommodation. You can see when they contract, the lens is much more concave or it's much more flattened when they're relaxed. And these are your ciliary muscles. Myopia, nearsightedness, they're focused in front of the retina. Elongated eyeball, the eyeballs are too long. Runs in the family. Everyone in my family and my kids, every one of them wears glasses. Corrected by concave lenses in eyeglasses or contacts, or again by LASIK. Hyperopia, farsightedness. Again, the image is brought to a point of focus behind the retina. The eyeball's too short. Convex lenses. Concave and convex. Astigmatism. What you have is an asymmetry between the cornea and the lens. And you can see how the light, instead of going back and focusing, it's going in different directions because of the change, the asymmetry, the change of shape between the cornea and the lens. When you test for astigmatism, we will have you look at a chart, a circle, with lines radiating out from the middle, and just like spokes on a wheel. If you don't have an astigmatism, you can see all those lines and they're clear. But if you have an astigmatism, kind of like in D there, some of the lines will look fuzzy or they'll look like they're going in the wrong direction because the light is being scattered as it goes through the cornea and the lens because of a defect there. Okay, the retina. The retina is in the back of the eye. Axons in the retina are gathered at a point called the optic disc. And when we do the lab, we will use an ophthalmoscope. Put you in a dark room, get you to look in. You can see, look at each other's eyes. You'll be able to see the optic disc in the back. You'll see the optic nerve. You'll see blood vessels coming in. It's kind of neat. Yeah, well. They come in through the optic disc. On the optic disc, there's no photoreceptors. It's a blind spot. Now, you never know it because your brain just compensates for it. But there is a part of your eye that does not pick up any information, and that's the optic disc. Photoreceptors. You have the layers of the retina, you have photoreceptors, rods and cone, are in the inner layer. Now this goes kind of against practical thought. You would expect that whatever's going to pick up the light is going to be on the outside of the retina, closest to where the light's coming in. But that's not the case. The photoreceptors, the rods and cones, are on the inner layer. They synapse with a middle layer of bipolar cells. A bipolar, remember, is a neuron that has more than one dendrite. It branches out, which synapse on the outer layer or the ganglion. So this is a direction of light coming down this way. Optic nerve fibers, bipolar cells, amacrine, ganglions. It's got to get through all that to get down to the photoreceptor cells. Why? I have no idea. But that's just how it is. But that's the retina, layers of the retina. Rods and cones. Remember, those are your photoreceptors. You have a lot more rods than you have cones. Outer segment of flattened disc with photopigment molecules. Here's your flattened discs down here. kind of see them here. These wear out, they break off, they are phagocytized by the pigment epithelium, and then they're replaced. Outer segment of flattened disc with the photopigment molecules, and then you have the inner segment with the receptor cells in that. So here's your outer segment. Here's your rod. Here's your cone. The inner segment's more the cell body nucleus, and your synaptic endings again are on the other end, on the inside of it actually. Retinal pigment epithelium. It's located under the rods and cones. It phagocytizes shed outer discs. It absorbs scattered light. It delivers nutrients. You don't have all the blood vessels in there. It helps suppress the immune attack in the retina. And it's participating in the visual cycle of the retina. We'll get into that. There are chemicals, rhodopsin, opsins, that are changed when the light hits, and they have to be recycled from the cis to the trans and back to the cis, and that happens in the pigment epithelium. And they help stabilize the ionic concentration so that you have the ions for a regular neural activity. Rods allow black and white vision in low light. They contain the pigment rhodopsin, which absorbs green light best. At night, you will see a green object better than you will see a red or a blue object. Simply because if it doesn't absorb the light, you're not going to see it. If it absorbs one color better, that's what you're going to see. Absorption causes rhodopsin to dissociate into retinaldehyde and opsin. Retinaldehyde is from retinol, which is from vitamin A. One of the first things you see with a vitamin A deficiency is poor night vision. And I know, my cousin was Navy SEALs, and they gave them, they kept them with vitamin A to give the night vision keep the night vision as sharp as they could. Absorption causes the rhodopsin to dissociate into retinaldehyde and opsin. The retinaldehyde from vitamin A is called the bleaching reaction, because once it dissociates, it's got to be put back together so it can work again. So when you're staring with the eye, and we'll see this in the lab, you'll have some bleaching action, especially in rods and more so with the cones also. Visual cycle of retinol. It exists in the 11 cis form. When to activate it, it needs to move into an all-trans form. Remember when we talked about the molecules? Cis, they're on the same side. Trans, they're on the outside. These are hydrogen molecules in this case. It must be incorporated back into the cis form to be able to be used again. This again happens in the pigmented epithelial cells. Bleaching. Bleaching is what happens as it's changed and then gets changed back. What you've got here are the hydrogens down here. These switching, I believe, is where it is, right here, between the cis and the trans. Okay, dark adaptation. Now, I always used to think, dark adaptation was simply it took that long for your pupil to dilate and let more light in. That isn't, I mean, that's part of it, but that's not really what happens. Person enters a dark room, there are fewer photopigments in the rods and cones. You go into the dark for about 20 minutes, the pigments are produced, and they're moved into the rods and cones, and that's why you can see so much better at dark. And I think they say it's like 100,000 times. The dark photoreceptors inhibit, okay, the dark current. Let's go back here. Person walks into a dark room, the light, there are fewer photopigments in the rods and cones, the rhodopsin. The body makes it, you kick more photopigments in, you pick up more light. Okay? Now the other thing that happens is what they call the dark current. And they're not quite sure really why this is doing it. But in the eyes, in the dark, instead of being depolarized, hyperpolarized, the eyes are depolarized. In the dark, sodium is allowed in. The gate's open. Sodium is allowed in. So it takes a lot less light to fire a neuron. As soon as you get light, it closes the sodium channels. It takes the GABA, cyclic GAMA, and the light breaks it down, so it closes the sodium gates. You don't get sodium into the neurons, and then they start to depolarize. It goes exactly the opposite of what you would think happens. You would think, anyway, let's look at this. The dark current. The dark current is a current of sodium, and it hyperpolarizes the bipolar cells. Again, the bipolar have multiple dendrites. It hyperpolarizes. You would think in the dark that it would depolarize and move them in the other direction.
Why would you think that? Because you let calcium in when you depolarize?
Well, at this point, it's just sodium.
Okay.
And sodium channels in the rods and cones are always open. Inhibit bipolar cells. The sodium channels are open, depolarizing the photoreceptor. Allows photoreceptor release, inhibiting neurotransmitters in the dark. That would just, it goes against all logic. And I'm not sure they're sure why it happens. When light hits the photoreceptors, the G proteins close its channels. The G proteins are called transducins. You get those in the nose as part of that, and in the eyes. The activation of the phosphodiesterase converts the cyclic gamma aminophosphate to gamma aminophosphate. This closes the sodium channels. Photoreceptors are hyperpolarized, and the inhibition of the bipolar cells. It is the bipolar. You would think that the bipolar cells are going to be kind of depolarized in the dark, and what happens is they depolarize in the light and they hyperpolarize in the dark. And I'm not sure they know exactly why that happens.
What about like nocturnal animals?
Nocturnal, I think in the mammals it still happens that way. It's just the number of rods and cones. And we get into that. What happens night and day, your rods on the fovea centralis, you have much more of your cones there. And the cones, each cone has its own nerve and its own ganglion. That's where your acuity of vision is. That's where your sense of vision, if you've got it focused there, that's where your acuity is. The rods may have 100, I think the average is 105 nerve rods per ganglion. So if a rod fires, your eye knows it's been fired, but it doesn't have a clue as to what's going on. It has to, it's a combination between the rods and the cones. But it's the number of rods. That's where your light sensitivity, your dark sensitivity comes from. And the animals, the dogs, the cats at night, it's because they have more rods. And they're colorblind, so they don't have the cones. But anyway, this is part of the second part of the dark current. And so when light hits it, everything the dark current shuts down, the sodium channels close, and the bipolar are actually the inhibition is lifted, and they can fire easier. Dark current, this doesn't clear it up a heck of a lot, I'm sorry. But,
there's another one later that comes, the figure 10.31, about this.
yeah, but you see the bipolar cells can now stimulate ganglion cells. That's in the light. So you're not using the bipolar cells in the dark because they're inhibited by the dark current. As soon as light hits and shuts off that cyclic GMBA, converts the cyclic GMP to GMP, it closes the sodium channels, it shuts down the dark current, and allows the bipolar cells to start sending stimulus back to the ganglion.
Yeah, they just show us how it doesn't work.
Yeah, they show us how it doesn't work, and I think when we get into the lab, I'll go over it with you again. Cones are less sensitive to light, but that's where we get our color vision. In the primate mammals, monkeys, humans, apes, we have trichromatic vision. Short wavelength, blue, medium wavelength, green, long wavelength, red. We pick up three wavelengths of light. And again, what happens when we see color is the wavelengths are coming in, they're hitting different receptors, and the axons are firing, they're going into the nerve, and the nerve's interpreting the color. The brain, excuse me. So here's the 400, the blue, the short wavelength is 380 to 520, the green is 450 to 620, the red is 480 to almost 700.
So in our notes it says that violet is the shortest.
What's that?
In the notes it says that violet is the shortest.
Violet is a blue, it's in the blue.
Okay.
That's the blue.
I'm backwards from everybody because that's the color I can see the best. Like, I'll see it usually in the first time.
You'll see it when it doesn't exist, the problem. Okay, instead of the opsin, remember the rhodopsin broke down from the retinaldehyde to the opsins? You have photoopsins. They vary in each cone. In your retina, you have the fovea centralis. That's where most of your cones are. That's where your visual acuity is. Again, each cone has a one-to-one relationship with the nerves. So when the light hits it, it's going into its own nerve. The brain can really tell what's happening. With the rods, you'll probably have about 100. With the cone, with the rods, you'll probably have about 100. Which allows more light in, but you don't have the acuity. Convergence of a lot of rods into a single ganglion increases the light sensitivity. You don't have the acuity. You have the acuity in the cones because it's a one-to-one relationship between the cone and the ganglion. Now, see what happens in the fovea centralis. Here's your optic disc. This is your retina. Here's your fovea centralis right here. The rods are kind of pushed aside, and the light is coming in directly on the cones. That's where you get your acuity. Retinal convergence and light sensitivity. Here, you have a lot. You have convergence because you have a lot of rods converging on one ganglion. In the cones and the fovea, they're individual ganglions. So what happens is you pick up the light better because you have more rods to pick it up, but your acuity, but because they're all coming into the same ganglion, they're all firing, you don't have a sense of visual acuity. You know it's there, but you can't really tell what it is.
So better acuity with cones.
The acuity, the clearness of vision comes from the cones. The light sensitivity comes from the rods. You need the rods to see at night, and that's why you don't see color at night. You see black and white. That's your rods. Neural pathways. The ganglion synapse on the lateral geniculate nucleus of the thalamus, and then it goes from the lateral portion of the retina does not cross sides, but the medial portion does. It's interesting that on your lateral portion it comes through, it doesn't cross over. Your medial portion will cross over in, I think it's in the optic chiasm. Okay. Point of view, eyes are focused. You see up there it has monocular, binocular. What happens is the fact that you have overlap. One eye, if I close one eye, I can see from here, try not to look too bad, from here to here. I close the other eye, I see from here to here. It's where you see the same thing from both eyes, the binocular vision, that gives you your depth of field. I've got a friend who hunts who lost an eye in an accident, and he's got to use a rangefinder because he cannot tell how far that animal or the target, is away from him. He just does not have any depth of field. So that's your binocular field that gives you that. Then your lenses comes in through the optic chiasm, the optic tract, superior colliculus. If that's destroyed, you lose your sight. For your sense of smell, it's the inferior colliculus. Here's your lateral geniculate nucleus. We'll talk about that in a second. And the occipital lobe is where the visual cortex is. Neurons from the thalamus, synapse on the striate cortex of the occipital lobe. Now, 20 to 30% of the ganglion cells axon, synapse on the superior colliculus, or the midbrain. This is right in here. You see some of them are going into the superior colliculus, the others are going into the occipital lobe. Let me hang on 1 second. I'm just looking for something. Okay, 20% of your ganglion cells synapse on the superior colliculus. that kind of tells you where something is out there. And another 70 to 80% go into the occipital lobe. That tells you what it is. Your brain is, you have two different areas. It's trying to decipher. One, you know something's out there. Secondly, it tells you what's out there. Eye movement. Now, it's produced by the extrinsic eye muscles. There's three types of movement. Now, what happens is you have the fovea centralis here. That's your sensitive part. What happens is, as the light is focused on that, some parts of it will pick up light right in the center, some on the edge. But what happens is you have the saccadic movement of the eye muscles, and it's essentially sweeping at about 800 times a second, I believe. The saccadic eye movement, high velocity movements, keep the image focused on the fovea centralis. So it's essentially, because the fovea doesn't, it picks up a pinpoint of light, what happens is the eye just sweeps the image across. And that's what gives your action potentials and your pickup of the image. The pursuit movement matches speed, pursuit movements. That's where your eyes match the speed of an object coming in front of you. So you have the saccadic movement, that's getting the image on the fovea centralis. Pursuit movement is watching something going by. The movement of an object, moving object. Divergence allows eyes to converge, so the image is at the fovea with both eyes. That's divergence movements. That allows the eyes to converge on an image so both fovea have the image. Ganglion cells have a photopigment called melopsin. That sends sensory images of the brightness of the light. It controls the irises. If the light's too bright, the irises constrict. It also, remember melanin out of the hypothalamus. It reacts to the amount of darkness. Where melanin is secreted, it slows everything down. It allows you to sleep. So you have melopsinin and you have melanin. Sets the circadian rhythms, light and dark. Wakefulness helps you go to sleep. Neural processing. Ganglion cell receptive fields. You have the ganglion, the areas of the retina with photoreceptors send input to the ganglion cells. Some of the ganglion cells, let me see if we've got some of the ganglion cells have receptive fields. If you put a spot of light into the ganglion cell receptive field, in this one, it'll stimulate it. In this off-center, on this ganglion cell, it will stimulate it when it's off-center. Like I said.
It's not that it turns on once the light is on it? No?
No.
Okay.
It's just how much it stimulates depending on where the light is in different ganglion cells. And this is, again, is what I was telling you. Kind of like the dark pathway, kind of like this. You have this antagonistic give and take. So depending on how much the off-center field is stimulated versus the center, it gives an edge to the objects. If the light's coming in, some of it on the off-center of the ganglion cell, some of it on-center, the brain interprets it as an edge of an object, and that's how you pick up the edges. You get a sharp edge vision. Light in the center stimulates light on the surrounding, inhibits the ganglion, light on the center, inhibits some of the other surroundings of ganglions. So some are excited, some are inhibited. The brain interprets it differently. Light across the center and surrounding is less effective at stimulating. It doesn't, you want the spots of light. So some ganglions have off-center fields, some of them have center fields. That's where they are excited the most. And the edge of the receptive field stimulates the ganglion so that you can get, again, the edges of your light and dark and objects. Okay. It's a lot. It's a lot.
It took a long time to photocopy.
When I look at this again, I want to look at kind of the take-home message. The take-home message is how the cones and rods work, how the difference between able to see in the dark, the difference in the different colors that you pick up. The blues are on one gene, the reds and the greens are another. It's an X-linked gene. So you have different color blindnesses varying on genders. Am I going to ask you to draw this picture of a ganglion or something like that, or the cochlea in the ear? No, I would kind of doubt that. But I want you to know that things like the ear, you're going to go in through the ear, you're going to hit the tympanic member, you can go into the oval window, you can go into the cochlea, you know what happens in that, and you know that there's inner and outer cilia. You've got the round window, you know that you've got the perilymph, the endolymph. The general aspects of it, like I said, it's kind of like me trying to make you remember the Krebs cycle. You'll never use it again in your life. I'd rather you pick up what you're going to apply as you go into nursing and as you go into what you're actually using in this class. So in the eye, you're going to need to know the light path. You're going to need to know what the fovea central is. You know what the optic is. You're going to need to know what the blind spot is. You're going to need to know the light path. You're going to need to know what happens with the iris, what happens with the ciliary muscles. Why? Why do we have presbyopia? Am I going to ask you to give me a detailed explanation of the dark current? No. But know that there is one. Okay? You want to take 5 minutes, or you want to head right into 11?
5 minutes.
Okay.
That's mine. That's my thought.
Or you don't want to head into 11? We're going into 11. And if we get, so that we can do 12 next week, we probably would have time to do a review before we, but we'll see. Hey, we get animations this time. Okay. Let's start chapter 11. We talked about endocrine glands. What's the difference between an endocrine gland and, say, an exocrine gland?
Endocrine gland goes into the system. Exocrine gland is like a sweat gland that goes out of the body.
Close. Endocrine goes into the blood. Exocrine goes into a duct. Okay? That's right. And then you have paracrine and acrine. Paracrine is autocrine, excuse me. Paracrine and autocrine. Paracrine goes into the tissue for cells around it. Autocrine goes into the tissue into its own cells. Self. That's an autocrine, producing hormones that reacts on the cell it's producing. Paracrine produces hormones that act on the cell around it. Endocrine, it goes into the blood, it goes to all the cells in the body. Exocrine goes into a duct. Sweat, you have exocrine glands in the stomach and the intestines, things like that. Endocrine glands and hormones. Okay, here you have the pineal, the pituitary, the hypothalamus, thyroid, adrenal, pancreas, ovary, and testes. Endocrine glands, they're ductless. They secrete hormones into the blood. The hormones are carried to the target cells having receptors for those hormones. You know, TSH is secreted, thyroid stimulating hormone is secreted by the pituitary. It doesn't do anything unless it's a thyroid cell. Unless it has the receptors, it doesn't do anything. But the TSH, thyroid stimulating hormone, is to virtually every cell in the body in, I think it's like 7 seconds. It doesn't take long. But it doesn't do them any good because they don't have the receptors. Many organs secrete hormones other than those discussed in this chapter. Heart, liver, kidneys, adipose tissue. And we will get into some of those. Adipose. Let's see here. Adipose, leptin from the adipose, it suppresses appetite. Adrenal cortex, this is one you probably ought to know. Glucocorticoids, aldosterone. Angiotensin, aldosterone system when we get into renal function. You're going to need to know that. Influence glucose metabolism. The glucocorticoids influence glucose metabolism. Aldosterone produces sodium retention and potassium excretion in the urine. This is all in your book. Adrenal medulla, epinephrine. Remember we talked about that in your fight or flight. It's part of the parasympathetic pathway of the adrenal medulla. Hypothalamus, it controls the anterior pituitary. It releases hormones that tell the anterior pituitary to release hormones that are stored there. Small intestine, secretin, and cholecystokinin. That's for inhibits gastric juice because you don't want to dump. It's from the small intestine. You don't want to dump a lot of active gastric juice into the small intestine, or it'll be rotted away. The stomach has the lining that gives the small intestine down. It is now. Islets of Langerhans. You know, I was going to think. I was going to tell you I was going to go to the islands on spring break, but it's not those. It was a bad joke. Kidneys, erythropoietin, very big if you're a chemo patient. Erythropoietin goes into the bone marrow, causes it and stimulates production of red blood cells, erythrocytes. When you're on chemo, that's pretty much destroyed. And if you have stock in Genentech, you're going to love it because they make Neupogen and Leupogen. Stimulates the bone marrow to make red cells and white cells, keeps from having to transfuse like we used to for cancer patients. And they're making them. Liver has Somatomedins, it stimulates cartilage. Ovaries, progesterone, secondary female characteristics. And in the male, female, parathyroid. Parathyroid hormone increases calcium absorption. If you don't have calcium, it increases it from the gut, it increases it in the kidney, and if you still don't have enough, it tells your bones to start breaking down. So, calcium is pretty important, and the parathyroid hormone is what helps regulate it. Pineal gland, melatonin, hypothalamus, anterior pituitary, affects secretion of gonadotrophin hormones. The melatonin also works on the limbic system, sleep, helps you sleep, your circadian rhythms. Anterior pituitary, trophic hormones, gonadotrophic, endocrine glands, other organs, stimulates growth, development of target organs, stimulates secretion of other hormones. Posterior pituitary, ADH, we talked about that, the kidney, antidiuretic hormone. If your osmoreceptors say your blood volume is down, ADH is secreted. It causes the kidneys to reabsorb water. You have less urine, it builds up the blood volume. Skin, vitamin D. And vitamin D works with calcium, absorption of calcium in the small intestine. So your skin is actually an endocrine organ. Stomach, gastrin. Gastrin is secreted. It activates the gastric juices. Testes, testosterone. Secondary sexual development. Prostate, seminal vesicles, and other male organs. Also, you do have testosterone in the female also. Thymus, white cell production. Thyroid, thyroxine, T4, triiodothyronine, T3, and that is metabolic rate in the cells. Growth and development and the metabolic rate in the cells.
Hormonal communication generally begins with a part of the neuroendocrine system receiving sensory information and reacting by issuing a command to the body in the form of a hormone. In this example, dehydration is detected by osmoreceptors in the hypothalamus, which then directs or stimulates the posterior pituitary gland to release antidiuretic hormone, ADH. The next step is to transport the hormone molecules to target cells. This is typically done via the bloodstream. Here, ADH is transported through the bloodstream to the kidneys and blood vessels. When hormone molecules reach target cells, they will bind to matching receptors on those cells, and the hormone receptor complexes will trigger changes in the target cells. This causes the kidneys to reduce the urine volume output, thereby increasing water retention and countering dehydration. In blood vessels, vasoconstriction is increased, leading to higher blood pressure and thus countering the blood pressure drop caused by dehydration.
Okay, chemical classification. Amines, tyrosine, and tryptophan. Hormones from the adrenal medulla. Adrenal, adrenal. On top of the kidneys. Your two adrenal glands are on top of the kidneys. Thyroid and pineal. What's from the pineal gland? Melatonin. Thyroid, thyroxine, and triiodothyronine, T3 and T4. Polypeptides, ADH, insulin, and growth hormone. Now, the thing about polypeptides, and we'll probably get it later, is these are protein-based. if you take them orally, they're digested in the stomach. Whereas your thyroid hormone and melatonin are not. That's why if you have an insulin-dependent person, you have to give it by injection. You can't take insulin orally. Now, you can take drugs that help insulin secretion, but not insulin. Glycoproteins, polypeptides, and a carbohydrate.
I'm sorry, but eventually when your nutrient don't get in contact with the blood, that affects protein.
I'm sorry.
We're talking about insulin back. You said that's
We're talking about insulin, yeah.
inject. And you said, why can't you ingest the insulin?
Because it's digested in the stomach.
Before it would
Before it
reach-
gets to do anything, it's
Okay,
digested.
okay.
Because it's a polypeptide.
Okay.
That's why you have to take insulin as a shot. Or as they have it now, pumps. Insulin pumps. Glycoproteins, polypeptides with a carbohydrate on it, by definition of glycoprotein. FSH and LH, sex steroids. Lipid, derived from cholesterol, the 5-carbon ring, cholesterol, testosterone, estradiol, progesterone, cortisol. They're secreted by the adrenal. There are only two places that secrete this. The adrenal cortex, which secretes the progesterone, cortisol, the testosterone, is mostly from the testosterone, estradiol, I guess, are mostly from the ovaries and the testes, though some is secreted by the adrenal cortex. Now you remember, your adrenal gland has two parts. You have the cortex and the medulla. The medulla secretes epinephrine and norepinephrine. It's part of the parasympathetic. It's part of the sympathetic system. And this is the adrenal cortex, not the medulla. Hormone classifications: polar, water soluble. Polar, like when we talked in chapter 1, polar molecules cannot go through cell membranes. They must be injected, or they bind, have receptors on the outer part of the membrane, activate a secondary messenger, something like that. Non-polar, insoluble in water, they're called either non-polar or lipophilic. They can enter the target cells. They can get through the plasma membrane. Your cholesterol, steroid hormones, thyroid hormone. They can get into the target directly. Steroid, thyroid, and take them orally. They're not broken down in the stomach, so you can take them in a pill. This is just the synthesis of the hormones, and I'm not going to worry about that tonight.
The non-polar can be taken as a pill?
Yes. Yes. Prohormones and prehormones. Remember when we talked about, the production of insulin, it started from, in the cells, from pre-pro to insulin. It started as a long chain that's broken up and it's made into a dipeptide, 2 polypeptide chains in insulin. Prehormones are inactive. The cells that use them have to modify them. Prohormones are inactive hormones that are cut and spliced together to be active. Insulin, remember, insulin came as a long polypeptide. It was cut, it was spliced into like a 20 and a 30 carbon chain. Prehormone, vitamin D in the skin, it's hydroxylated in the liver and kidneys. Testes, testosterone, and estradiol, again, they're pre-hormones. They have to be changed in the cells that use them. Thyroid, thyroxine T4 has to be changed into T3 to be used. Hormones are like neurotransmitters. They interact with specific receptors. They cause changes within the cell. They turn on and off cell activity. They turn on activity. The insulin turns on cell activity. Gets the cell to absorb glucose. Thyroid turns on cell activity to get the DNA in the thyroid gland to produce thyroid hormone. TSH does that also. Hormone interactions. The target cell may be responsive to several different hormones. Again, you have this antagonistic, depending on the volume, depending on the type of hormones. You have an antistic, synergistic, or permissive. A lot of the time it's an antagonistic one, and that's how you get this fine regulation again. Antagonistic, they're working. Synergistic, they work together. For instance, epinephrine and norepinephrine, they're synergistic. They add to each other. So if you have a heart, the contractions are picked up by epinephrine. If you have the norepinephrine, it picks it up even more. Permissive, one hormone allows the other hormone to work. Estradiol and prolactin. Estradiol allows the prolactin to work for the secretion of milk. I believe that's how it goes, and we'll look at that. A cell response is dependent on the amount of hormone or the combination. And we talk about, okay, let's just go through this. Synergistic. They produce two or more hormones produce a particular effect. Epinephrine and norepinephrine. They affect the heart in the same way. And they're synergistic, so it's just a greater effect on the heart. Complementary, when each contributes a different piece of an overall outcome. Milk, estrogen, prolactin, and oxytocin. Oxytocin and estrogen help the prolactin, work with the prolactin for the excretion of milk. Permissive, when hormone makes a target more responsive to a second. Estrogen makes the uterus more responsive to the progesterone in pregnancy and implanting the follicle. Antagonistic, they work in opposite directions. Insulin causes the cell to absorb glucose, to take in glucose. Glucagon causes it to excrete glucose. Insulin stimulates fat storage, glucagon fat breakdown. They're antagonistic, they work in opposite directions. Half-life. The hormones are broken down very quickly. If you have a hormone in there and you're not breaking it down, this action is just going to build. So the body breaks them down very quickly, mostly through the liver. One that doesn't break down real quickly is thyroid. It can take weeks for it to hit its half-life. That's why if you've had a thyroidectomy or if you've irradiated the thyroid, it takes a while for those hormones to go down. Tissues respond when hormone concentrations are at normal level. Normal level versus a pharmacological level. Did they talk about that? Mm-hmm. Yeah. Pharmacological is where you're taking a bunch of them. A normal level of vitamin C may be what you get from your diet. A pharmacological level is at 4,000 milligrams. They tell you to take to fight a cold. That's the difference. High concentrations, when you get enough hormone there, it may bind to receptors that it's not designed to bind to. And I think one of them, that you look in the steroids for bodybuilding, the androgenic steroids, the brain tumors, the impotence, the problems because of the high concentration of hormones and steroids the weightlifters are taking to build bulk. They're just, it's not worth it. But what's happening is the hormones are in pharmacological concentrations. They're binding to areas they're not supposed to be binding to. Priming effects, upregulation, downregulation. Some target cells are going to respond to a hormone, and some ways they respond by increasing the number of receptors. Now, if you remember, in the plasma membrane, receptors are not always in the membrane. They are, and they're not always in one area of the membrane. It's like your synaptic receptors are in the ends of the synapse. They're not scattered along the sides of the neuron. And the body has the ability to insert receptors into the membrane as needed. And this is what's happening here. Then the receptors are there, it makes it more sensitive to the subsequent release. And if you have a minimal exposure to the hormone, the body's saying, I need to get every bit of this, so I'm going to put more receptors out there so that any hormone that's coming by, I'm going to grab and make use of it. On the other hand, you'll have a downregulation if you're, say you have a Graves' disease or Cushing's disease, where you've got all these hormones being pushed out. Your body will try to downregulate and will decrease the number of receptors. Adipose tissue in response to high concentrations of insulin. The body's going to try to protect itself from these high concentrations of hormones. So it downregulates, it cuts down the number of receptors. And that's why a lot of your hormones are not secreted constantly. They're secreted in spurts. You've got them when you need them, but there isn't a lot of it hanging around. We see that with thyroid stimulating hormone. Depending on how much thyroid you need, the level goes up and down. Mechanisms of hormone action. They bind to receptors on the target cells. The binding is highly specific. It's like the enzyme. Very specific on what it's going to bind to. High affinity. Once they bind, they're there. Low capacity. You don't need a lot of it. Okay, lipophilic. Non-polar hormones. Lipophilic. And thyroid hormone. They're not dissolved in the water. They're lipophilic or hydrophobic. So they're attached to a protein, thyroglobulin for the thyroid, and it carries it and it releases it at the cells that are going to use it. And it disassociates from the carrier and then it diffuses across the plasma membrane. So we'll use thyroid as an example. It travels on thyroglobulin to the target cells attached to the carrier protein, which again is thyroglobulin. At the target cells, it disassociates and diffuses across the plasma membrane. It gets into the cytoplasm of the cell. It goes right across the membrane. Now, some receptors are in the nucleus, and the nuclear hormone receptors. Some of the receptors are in the cytoplasm. They bind to the hormone and then carry it into the nucleus. It can go either way. Hormones that bind to the receptors in the nucleus. The hormones activate genetic transcription by serving as transcription factors. Now I'm going to ask you to go back. We're talking about chapter 2 now, transcription factors. Something like that. Too long ago, huh? Too many cells have burned up in the meantime. But as I remember, I talked about thyroid stimulating hormone. It's secreted, it gets into the blood, it gets back to the cells, gets through the plasma membrane, it gets into the nucleus of the cell. It turns on in the thyroid cell, it gets in, it starts transcription of thyroid hormone which is, you start with the pre-mRNA, then the mRNA. The mRNA carries it out to the ribosomes. The ribosomes produce the protein. They're packaged by the Golgi bodies. The Golgi bodies get them to the cell membrane and excrete them into the blood, where they go to other cells that need thyroid hormone. The TSH doesn't work on the other cells because they're not thyroid cells. It works on the thyroid cells. It gets into the nucleus. They then produce new proteins that are excreted. And it's kind of like making enzymes or whatever else that cell is supposed to make. In the thyroid cells, it's thyroid. Two regions. The ligand binding domain and the DNA binding domain, the nuclear and the nucleus. The binding of the hormone activates the DNA binding domain to a hormone response element on the DNA. In other words, there is a codon. There is a code on the DNA that this hormone is going to bind to, and it says transcribe. Open up the mRNA, the pre-mRNA in, cut out the introns, cut out the exons, make mRNA, and ship it off to the ribosomes. Binding the hormone activates the DNA binding domain to a hormone response element. This is what would happen if your TSH got into the nucleus of the thyroid. And they're saying that there's ligands, there's receptors, but they don't always know what, for the different ligands, they don't know what the ligand is, so they call them orphan receptors. This is something that's really kind of on the edge, trying to figure out what they've got, 60 or 70 of them named so far. But you have the ligand binding domain in the nucleus and you have the DNA binding domain. Steroid hormones. Their receptors are in the cytoplasm. After binding, they translocate to the nucleus and bind to the DNA. Should have one. So here, you've got the blood, you've got the carrier protein, you've got the receptor protein. Once it with the receptor protein, it goes into the nucleus and opens up the DNA, and you start the mRNA protein synthesis and steroid hormone response. Usually these are all mostly controlled by a negative feedback system. Hormones respond to elements in the DNA. Steroid hormones respond to elements. They have two half sites. Two ligand-bound receptors must bind. It's called dimerization, and they form a homodimer. In the DNA, the DNA splits up. You've got two sites that these ligands have to bind. And sometimes they're the same ligand that binds. That's why you have a homodimer. Dimer 2, homo the same. With thyroid, you have two sites that it has to bind, but they're two different molecules. And they're called, it's still dimerization, but it's called a heterodimer. Steroid. it gets into it gets to the binding site. It gets into the nucleus. You have these two half sites to get to the target gene starting to replicate. The dimerization of the receptor, you have the two ligand-bound hormones. They bind to the DNA, and then that's like the start of the genetic transcription. That's what tells the DNA to split open and let the mRNA in. That starts your genetic transcription. These, because they are the same, are a homodimer.
Steroid hormones are not water-soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
And the proteins are packaged and either used by the cell or sent out into the bloodstream to be used by other cells. Okay, you have coactivators or copressors. And this is just like a coenzyme. It's a molecule that attaches and allows the hormone, allows it to start the transcription. They bind to the nuclear receptors in specific regions. It changes the effect of a given hormone on different cells, depending on the nuclear or what the cell is supposed to be doing. Thyroid hormone. T4 travels to the target cells on TBG, thyrobinding globulin. Inside the target, it's converted to T3. Receptor proteins are located inside the nucleus bound to DNA. So here, excuse me.
Would you go back to the other slide?
Sure.
The thyroid hormone?
Thyroxine T4 travels on thyrobinding globulin. That's the carrier protein. Your active part of thyroxine is a T3, triiodothyronine. Receptor proteins for the conversion are located in the nucleus found in the DNA. Okay?
Thank you.
So here, carrier proteins, TBG. T4 binds, it's converted to T3. T3 goes in and binds in the nucleus, causing, again, transcription, protein synthesis, thyroid hormone secretion. And like I said, in thyroid, you do not have a homodimer. You have a heterodimer. The cis-9 retinoic acid and the T3 are the two that you have. So they bind, instead of having like we had the two steroid hormones in the homodimer, you have two different ones. Cis-9 retinoic acid and triiodothyronine. Now, notice on this, iodine. You absolutely have to have iodine in your diet. to form the thyroid hormone, to form the active form. If you look in the book, I have it here.
Thyroxine is a...
We'll get it to in a second. Japan. They're handing out millions, if not hundreds of millions, of tablets of potassium iodide. The radiation that's being spewed out from nuclear plants attaches to the iodine in the body and is picked up in the thyroid and is there to cause cancer. What they're doing is they're saturating the iodine in the body with potassium iodide so it doesn't pick, the radioactive iodine isn't picked up. That's why they're handing out millions of pills. I mean, people over here are starting to take them too, which is kind of ridiculous. But that's why, when you hear them, what they do in case of radiation, they hand out iodine, potassium iodine. That's why they're doing it, because it's all picked up. The iodine is picked up in the thyroid. They want to saturate the thyroid with non-radioactive iodine, and then not allow it to pick up the radioactive iodine.
So water-insoluble hormone that is brought to the target cells via a protein carrier. Because it is lipophilic, thyroxine can easily pass through the cell membrane. Thyroxine contains 4 iodines and is often abbreviated T4, tetraiodothyronine. The thyroid gland also secretes smaller amounts of a similar molecule that has only 3 iodines, called triiodothyronine, T3. Both hormones enter target cells, but all the T4 that enters is converted into T3. Thus, only the T3 form of the hormone enters the nucleus and binds to nuclear receptor proteins. The hormone receptor protein complex, in turn, binds to the appropriate hormone response elements on DNA. The binding of the hormone receptor complex has a direct effect on the level of transcription at the site where it binds. The messenger RNA, mRNA, produced then codes for specific proteins.
Which again are secreted out. Hormones that use second messengers. Remember, we have the G proteins, hormones that cannot cross the cellular membrane, so they bind to receptors and stimulate a secondary messenger. Tyrosine, phospholipase C, adenylate cyclase are 3 that have been shown. Adenylate cyclase or cyclic AMP is part of the cyclic AMP system. You've seen that as a secondary messenger in many different cells. Used by epinephrine and norepinephrine, it binds to the beta-androgenic receptor. G protein dissociates. Uses ATP to make CAMP. Activates the protein kinase. Kinase, by definition, phosphorylates. Kinase, so anytime, and it's a reasonable test question.
What is it?
Kinase, by definition, its activity, a kinase is an enzyme, it ends in ASE. By definition, it phosphorylates. So creatine kinase phosphorylates creatine. Glucose phosphokinase. You don't need phospho anymore. Glucose kinase phosphorylates glucose in the liver, doesn't let it out. Keeps it from going across membranes. Things like that. Protein kinase phosphorylates proteins in the target cell to alter cell metabolism. Phosphodiesterase breaks down the CAMP. Again, this uses energy. Using ATP to make this work. So here you have receptor, again, stimulating the G proteins, alpha, beta, gamma. In this one, it says the alpha, along with the adenylate cyclase and ATP, is making a CAMP. Adenosine triphosphate is broken down to adenosine monophosphate. Cyclic adenosine monophosphate, CAMP. It's attaching to the protein kinase, it's activating it, and this is phosphorylating proteins, activating enzymes or inactivating other enzymes depending on the cell. And the regulatory subunit again is the CAMP for negative feedback if need be.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of CAMP from ATP. The CAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
See what it means by phosphorylase? When glucose gets into the liver and it's formed into glycogen or glucose 6-phosphate, by attaching the phosphate with the glucose and phosphate attached, it cannot get back out across the cell membrane. So you don't have it leaking out into the bloodstream. With a phosphorylase, takes the phosphate group off, that allows the glucose to secrete, it allows the liver to secrete glucose. In the fight or flight, your glucose goes up. The epinephrine is part of the fight or flight. You want as much sugar in the body to feed muscles or whatever else you need fed. Generally it's muscles, hard muscles, skeletal muscles, in the fight or flight. That's where the epinephrine is stimulating the phosphorylase, eventually, the phosphorylase and the release of glucose. Okay, phospholipase C. I think this is just pretty much what we went through. Binds to the androgenic receptors, G protein activates phospholipase C. It produces an IP3 and getting awful late.
Yes.
Give me another 10 minutes. Bottom line, it activates calmodulin and activates protein kinases. Protein kinases do what? Phosphorylate. By definition, kinase phosphorylates.
So that's all we're really noticed to?
I'll get back to you on the IP3 and DAG. I'm just drawing a blank right now. Okay. G proteins, again, secondary in the endoplasmic reticulum. This is especially in muscle cells, things like that. The endoplasmic reticulum, and on some of the hormone the bitter taste, the bitter sensation and taste. This is how this would work, because the calcium for that is being released from the endoplasmic reticulum. So that's phospholipase.
I'm sorry.
When we went through the sweet, salt, sour, the bitter, in the bitter, the neurotransmitter is activated by calcium being released from the endoplasmic reticulum. Phosphate C, I think we just went through that. Epinephrine, beta-androgenic or alpha-androgenic, we still get the calmodulin. And then this one is the liver, so the glucose gets the two methods for epinephrine. Beta-androgenic, it binds, you can't get across the membrane, you get the cyclic AMP, activated protein kinase, the glycogen, glucose 1-phosphate, glucose 6-phosphate, phosphatase takes the phosphate out, and glucose goes out. The second is the alpha-androgenic as opposed to the beta-androgenic. So you get the alpha effect of epinephrine, you get the calcium, the calmodulin, and then you're back to the protein kinase, which takes you back to the glycogen, glucose 1-phosphate, and glucose again. You have two methods of epinephrine acting on the liver. Tyrosine, insulin. Hang on a second. Wouldn't want to have to start over again. You know,
It gets.
I'm tired.
4:30, right? Isn't that what you said?
My day started at 4:30. So, we will start with tyrosine on next Tuesday. And then wherever we end up next Tuesday, we'll probably, if not through chapter 12, the end of chapter 12, we'll test up to that point on the 20th.
On whatever the
On the
first
12th.
April,
On the 12th. I was going to say the 22nd, and then do that with.
That's fine.
Is tomorrow next week or last week before the?
Next week is the last week before spring break.